All right, I am Will Serber, and it is my great pleasure to be introducing this section and giving you a little bit of background before turning it over to friends from OpenAI and Pacific Northwest National Labs. The title of the talk here, Designing, Deploying, and Scaling Autonomous Labs with OpenAI and PNNL. I'm the head of automation at Ginkgo and have been there for a handful of years. As a disclaimer, of course, this is required. I've been in Ginkgo for about 3 years, going backwards on this. Before that, I was head of automation at Zymergen. Before that, I was a commercial photographer for 5 years, not very helpful for my current career. Before that, I was an astrophysicist for a while, again, not the most helpful. But it's been fun combining all these things as much as I can into one interesting career. I've got a dog in the bottom right-hand corner named Soba, and that's her looking very bored in our workshop where we make racks. So the mission of the company is to make biology easier to engineer. And we've got about 400 or so employees, a little north of that. And we're expecting about 200 million in revenue this year, just to tell you a tiny bit about Ginkgo. And we're really focusing more and more on automation. So the talk is organized really, there are 3 fundamental segments to it. One, just a description of our belief that autonomous labs will transform biotechnology. Second, I'm going to tell you a little bit about what we think those labs might look like. And then third, just a super quick word on kind of how we go to market with that in practice. Starting with the first. So this is an image you might have seen Jason, our CEO, show off, but I do quite like it. So this is an old ad for IBM in which they were saying, one of their computers will place 150 engineers with slide rules. So there's a couple of reasons I really like this. One is, very clearly this is, someone might look at this and think like, oh my God, our jobs as engineers are at risk as a result of this technology. But we all know that there's way, way, way more software engineers today in the world than there were back then. So that's the way I feel about automation in the lab space too. And so it's nice to see this fantastic example. The other thing that's kind of interesting, sort of represented on this slide, is that these two things are not really equivalent. The slide rule is extremely flexible, but not automated. You need a human to run it. It feels similar to a pipette in that sense. And then in the top left-hand corner, you have the things that IBM was selling, which at the time were not particularly general purpose computers, but they were heavily automated. And it was really the advent of something in the top right-hand corner, the personal computer, that transformed the world and is why we have billions of computers in the world today. So that high automation, high flexibility is kind of the golden spot to be, where you can really have a transformative effect on the industry. Continuing this analogy a little bit to transportation, you can imagine trains or subways being in the top left-hand corner again, where totally automated, but you have to go where the rails are laid, or a car in the bottom right-hand corner. And this is the way the world has been for a long time. Like you basically have these 2 choices. And we're starting to see the emergence of something in the top right-hand corner. Waymo being the best example where within their constrained environment, it is both autonomous and flexible. And it will be interesting to see how this changes the world and the way that cities are laid out and people lead their lives. I suspect this will be quite a transformation as well, which is also why so much money pours into that space. In the lab space, we again have things in these 2 corners. We have the lab bench, extremely flexible, but not a lot of automation, no automation. And then you have the traditional work cells in the top left-hand corner filling this out. And to date, there's basically been nothing in the top right-hand corner. And so really what Ginkgo is going after is that top right-hand corner. We don't view ourselves, even though we have lab equipment, that's what we're selling, we don't view ourselves as competing fundamentally with the work cell per se. It's competing as much with the bench as the work cell. We are very much looking to transform the way that people do science in the way that the computer did, or the way that perhaps autonomous driving will. So this raises this question of, okay, how do you get there? How do you actually build that? Part 2. So one of our key beliefs is that an autonomous lab must allow for the same work you do in a traditional lab. So you need to be able to do all those same things. You can't be leaving out key parts of the workflow. This is the wordiest slide, I apologize for this one, but there are basically 6 things that we think are critical to replicate to get to that top right-hand corner. So we think you need reliable liquid handling that is easily programmable. We call this one out just because liquid handling is so much at the core of so many different workflows, and so it feels like it's worth special mention. Number 2, you need reliable material transport to any device. The reliable part is the hard part there. Like, lots of people have robot arms, but they're not really reliable enough to get to where we want. Number 3 is parameterized control of devices. You need, a good API. You need to be able to do many things with the device and get kind of full control over it. 4th is you need support for as many devices in a single setup as you would have in a traditional lab. And so we think, you don't want to have lots of different work cells with humans walking between them ideally. You don't want to be missing pieces of equipment that are on the bench in the idealized form of this. Fortunately, there's value to be gotten along the path to this vision, but this is the ultimate vision. 5th, support for parallel work. So if you imagine a traditional lab with a bunch of grad students or RAs running around in it, like, they are coordinating constantly to share equipment and to make the best use out of the capital expenditure. And so we believe that really to achieve what we want to achieve in the lab, you need to support tens to hundreds of parallel protocol requests from scientists, and critically ones that change daily. It's not the same 10 protocols they're going to be running over and over again. They're going to want to push the frontiers of what is possible and to be running different protocols. And then lastly, it should say an experience as easy for scientists to use as the bench. So we think we really, really need to lower the barrier to entry to using these tools. I'm going to tell you just a little bit about our solution. If you're excited to see more, you should come to our booth. It's the big pink one in kind of the middle of the floor. Very Valentine's Day themed. So we've made modular hardware, and we've kind of wrapped each instrument in hardware to make it more reliable and more robust so that it really behaves as well as possible. And we put all the accessories needed into this modular sort of Lego building block. You can put multiple racks, we call them, reconfigurable automation carts together, and they plug together kind of like Lego, and they just work. And so again, it's like this focus on reliability and robustness. You can start very small, as in the top left-hand corner, with just a few racks. So it's a small investment, begin automating at that scale, and then expand over time in a kind of nice linear way to add capacity or capability. And you can get to a very large system. I don't want to steal too much of Todd's thunder, but that system on the right is a 97 rack system purchased by the Department of Energy, by PNNL. And so this technology really scales from small to large. And then the other part that we've really invested a lot in is our software. And there's a ton to be said about this. We've tried to make it super easy to use, by which I mean writing protocols, launching protocols, monitoring them, getting the data off. But the one thing I wanted to emphasize in this talk, in this small intro, is just the interleaving of protocols. And so we've got distributed plate transport, and then we put a lot of time into our constraint solver that is actually scheduling the work. And so the cool thing here is the different colors of the bars up there, it's basically a little Gantt. The different colors are different protocols running on the same system at the same time. So run by different people, totally different workflows, requiring different instrumentation. And it figures out how to schedule it and to get the maximum utilization out of your instruments. So here is just a clear example where that Gantt, you can see there's a whole bunch of colored blocks all in the same vertical line, which means multiple things are moving simultaneously on the system. So you've got multiple robots transferring plates at the same time. You can also have one rack in error and the rest of the system keeps running. So you get very high availability. If there's a second identical rack, it'll fail over automatically. And then this is just the dark mode version of that same sort of Gantt. I'm sorry it's so dark here, but I love this because this is a screenshot of the scheduler in our system in Boston from just a few days ago. And this is it running 35 simultaneous protocols from different users. There are vendors out there that can run one or two things at a time, but nobody is approaching this level of concurrency. And I really think that allows you to make a core lab, an autonomous lab, that is much more effective and efficient than you could make with any other technology. This is our lab just down the street. If you want a tour of this, you can come to our booth and we'll give you a Uber coupon to just drive over there. And you can go there and come back in about half an hour and get a quick tour of our facility and then be back at SLAS in no time. So again, we'll pay for the Uber. Also on Thursday, we're going to be doing tours all day. So you can just show up at any time. 25 Dry Dock Avenue is the address. So please come on by. It's one thing to go see our booth. It's another thing to see the actual racks in the lab doing real science. So this is the fastest part, I promise. We are commercializing this in 2 ways. One is we are happy to build out autonomous labs, in your lab, and that looks like a traditional automation sale, or you can take advantage of the same technology via our CRO services. So we will run protocols for you if you want. There is a QR code for my LinkedIn if you want to make a connection. Happy to connect with more folks who are interested in this. Again, our booth 823, stop on by. That is my last slide, and I will leave it up for just another second as I see people scanning the QR code, and I will now move on. And then you'll have to find me afterwards, sorry. It is my pleasure to pass things over to Joy from OpenAI, who's going to tell you a little bit about some of the closed loop science we've been doing together on top of the platform I just described. Joy? You can use that, or you can just use the arrows.
Okay. All right. So I'm really excited to tell you more about this project that we did with Ginkgo over the last 6-ish months, where we basically gave GPT-5 control over Ginkgo's rack system, and we were able to make more protein, in this case SFGFE, at a lower cost than the previous human state-of-the-art result. Okay, so just a little bit of background. I think this was not quite as obvious 6 months ago as it is now. I generally think of AI and science kind of as this different stages of capability. So initially, we think of AI as tools. And this is commonly things like AlphaFold, both like general biology foundation models. And OpenAI also had our own paper out a few months ago about GPT-4b, which is actually just a very tiny model that we fine-tuned to generate Yamanaka factors variants, and this actually showed that there was like a 50x reprogramming efficiency. So this was like a interesting project kind of in the AI as tools regime. And then we see kind of AI evolving as tool users. So we start giving our models access to internet, access to computers, and we generally see a huge capability jump in this regime. And then the thing that I really want to talk about here is kind of AI as co-scientists working alongside humans. And then also in some cases, like in this project, designing experiments independently. And at the start of this project, GPT-5 had actually just finished training at OpenAI. And we knew that it was very knowledgeable. We knew that it was very good at programming and math and physics. But we haven't really tested it in biology. And part of this is because biology requires wet lab to really validate. So I think computational validation of computational results is not quite as interesting. And so with Ginkgo, we had this opportunity to really have the model design real biology wet lab experiments, and we can actually run them in the rack system and really kind of evaluate GPT-5's capability at doing real biology. So the experiment that we settled on is cell-free protein synthesis. So I have a few slides kind of talking about why this is important. Apologies to those in the audience who know way more about this than I do. But generally, protein production is pretty foundational to pretty much everything, including medicine and research. But generally, traditional methods are very slow and complicated. So you have to introduce DNA encoding a new protein into cells. Often you have to create a stable cell line, scale production in bioreactors, and this often takes on the order of weeks to months. So the limitations here is that it's often slow and inflexible. There's limited protein types that you can make. So for example, if you have a protein that is toxic to bacteria, you can't really grow it in bacteria. There's potential safety risks involved, and it's not quite portable because you require sterile facilities and large bioreactors. So cell-free protein synthesis kind of turns this regime inside out in some ways, where now instead of making proteins in living cells, you actually lyse the cells, so all the protein-making machinery inside the cells is now kind of free-floating in this tube, and you can actually just spike in your DNA construct, encoding a protein of interest, and make it this way. And so this is really good for kind of prototyping large libraries of different protein variants and just general high throughput workflows. And so what this can give us is faster drug discovery, personalized therapeutics, and we can get significant speed ups because now instead of having to make these cell lines, you can get proteins on the order of hours or days. High throughput, you can make many things in parallel. It's a lot more flexible and safe because now you don't have this kind of like contamination concerns. And the really nice thing here too is that the setup itself is not super complicated from a protocol perspective because it's kind of, at least from the model's view, right? So you add a bunch of different volumes of reactants into a tube, and then that tube just incubates. And so this is a good problem for our models because it doesn't require that much understanding of the physicality of the experiment. It's really just kind of thinking about optimizing the volumes of reactants that you're putting into the tube. So there's a nice kind of constraint on the action space, but there's still a lot of knobs for the model to tune and explore. Okay, so this is kind of the general setup. On top we have GPT-5 that is kind of doing the data analysis, reasoning, generating hypothesis. It's actually writing code to define the next set of experiments. And I'll kind of like walk through what the experimental design looks like a few slides out. But essentially this is a pydantic object that is like serialized into a JSON file that we're sharing between the two labs. And then from the Ginkgo side, the physical experiment is actually executed. So we have the automated liquid handling, the incubation, the fluorescence measurements, the standardization, all of these things are happening there. And then the metrics and the data and any kind of metadata about things that may have happened during the experiment, like for example, there's equipment that errored out on specific wells. All of that data is then sent back to GPT-5, and then we just kind of run this loop over and over again. So over the last 6 months, we ran around 36,000 different reactions across 580 384 well plates. So alongside the experiment designs, we also asked GPT-5 to kind of write lab notebooks and to document its kind of biological reasoning throughout this process. And part of this was because I wanted to monitor the model to make sure that it was actually doing something sensible. Because one boring case you can imagine is that the model just kind of does a dumb grid search and submits that as a plate design. That's not very interesting. So this was actually an excerpt from the first random lab notebook that I opened. The part that is bold, the NMP supplementation was in situ phosphorylation. This suggestion was actually the subject of a recent paper that was published around the time of our collaboration. It came from the Jewel lab, and they had achieved a huge reduction in the amount of cost per gram of protein that they were able to get from a cell-free system. And so the really interesting thing here is this bold line is actually one of the critical insights that they had in this paper. But GPT-5 was not trained on this paper because this paper had just come out. And also in the setting, GPT-5 didn't have access to internet. And so this is actually just from knowledge that was stored in the model weights. And so this was actually the initial sign of life to me that the model had enough biology knowledge in its model weights to actually successfully do this experiment. Something early on that we also had to figure out too was on almost every rollout, GPT-5 was complaining about batch effects and kind of just general variability between replicates. So as we all know, biology is very noisy. And so for each experimental condition we were testing, we were doing them in quadruplicate. So there were 4 wells kind of in random positions on a 384 well plate. And GPT-5, which is always complaining about, the same experimental condition from plate to plate is like not really consistent, and then it's not consistent kind of within plates. And so there was actually a huge amount of work from the Ginkgo team to reduce the coefficient of variation to something that was much more usable. And then at steady state, we were testing around 10,000 conditions per week. Okay, so these are the results that we got out of the model. The overall objective is to reduce the cost per gram of protein that we were making in this system. And we chose this objective because I think just going for titer itself is not that interesting because you can imagine just kind of throwing all the most expensive ingredients into a tube, and that's not really a scalable system. So here we have the first round of experiments that GPT designed. The horizontal axis is the amount of titer that we were getting out of these reactions in 3D 4-well plates. That's like grams per liter of protein as measured by fluorescence. And on the vertical axis, we have the reaction cost of each one of these reactions. And the star here is the soda 3D 4-well result that came out of the Julia lab. So this is actually a really timely paper for us because then we kind of had the human gold standard to benchmark against. And then in this setting, GPT-5 was just reasoning. It was given a prompt about what cell-free protein synthesis roughly is. It was given the pydantic model spec. And we just told it to write code to design 78 different experiments for this like 384-well plate. And we didn't really give it any information. And it didn't have access to papers, it didn't have access to internet. It was just kind of thinking in a closed box. And so these were the first designs that I came up with. I remember at this point, we were kind of happy that it made protein at all, because I know that if you kind of gave me, put me in a closed room and told me to write a reaction, I don't think I would have gotten results as good. But that being said, kind of looking at the plot on the right where we have the minimum dollar per gram that we can get, we're still, I think, around 14x away from the soda. So there was still quite a bit of room to go. Also on this step, we noticed that the model was kind of suggesting new reagents that we should add. And so we actually had the model rank all the reagents that it suggested we should add to the pydantic reagent list. And then we ranked them, and I think we took around the first 25-ish of those those reagents. And for the next step, we gave it to the model. So in general, we basically want to move in the direction of this blue arrow. We want to see this kind of Pareto frontier move to the right and downwards, meaning that we're getting more protein, but also the reactions are getting cheaper. So this is the next step. We see a decent amount of improvement, but it's not huge. We're still many orders of magnitude away from soda. And the difference here is on step 2, the model, so as we were designing these experiments, the model was able to use the new reagents. And so we see that step 3, we get quite a bit better, but we are kind of hitting, so as you go to the left, we see that the model is kind of not able to go down further in terms of the cost. And we also want to move a little bit further too in terms of titer. And so at this point, we thought, okay, we know that the model is able to make these incremental improvements just kind of based on knowledge in its model weights. But this is not how a human scientist will work. We would be able to read all the previous literature. We would be able to search the internet. We have access to a computer to kind of do different kinds of data analysis. So we thought, let's just throw all these tools at the model instead of kind of doing this artificial evaluation of how much internal knowledge the model has. And so for the next step, this is what tool use, and we see this huge jump. So we actually immediately exceed the 384-well soda, state of the art. And so now we actually kind of have to zoom in this region. So for the next part, we're kind of switching from log scale now to linear scale to make things a little bit easier to see. So the tools here, the model has access to its computer and a browser, and we see kind of from steps 3 to 4, the model gets better, and then step 5, the model gets even better. And so we can also kind of see the progress on the right as well, where we're kind of just getting much better with each step. So overall, when we retested these reactions in 2-ml tubes, where now we kind of have more oxygenation due to the better geometry of 2-ml tubes, we see a 40% reduction in the total reaction cost. And then when you look at the reagents alone, it's actually 57%. So it was a pretty decent achievement, I think, for the model, where from the beginning we weren't sure if we could design even a single reaction. The other interesting thing here too is that because we were optimizing at the 384-well plate level, I'm not sure how easy these numbers are to see, but you can kind of see the RF off, which is the result that came out of the Julia lab with the human gold standard. It's actually much lower in titer at the 384-well level than most of our reactions. And I think this is just because the model is designing experiments for this kind of low oxygenation condition in the 384-well geometry. And here I'm just kind of showing you some concrete examples of what the code the model actually saw looks like. I believe this is actually open source, so you could also browse this online on GitHub if you want to. But yeah, we basically just constrain the action space of the model so it can focus on kind of the biochemistry and not what's physically executable on the racks. So it was just sending these plate objects kind of back and forth between Ginkgo and OpenAI. And the plate has a list of sample objects. It has kind of the results of the experiment. And also you see there are things like reagent flags of what, if like a reagent is out of stock. Also metadata on different events that might have occurred during the execution of the experiment. And then we also have validation for all the different parts of the plates. And this just makes sure that, for example, like a plate has the right number of samples, it has a sufficient number of replicates. And what this gives us is that we know that if a plate passes all these validations, then this experiment is executable on the rack system. So this gives us a really nice way to kind of programmatically check all the rollouts and the outputs are coming from the model without trying to run it on the rack and then only later to realize that it was broken. Let's see, I have a few more of these where, we're validating that the reagents are available. We're validating that a model can only add things in 25 nanoliter increments because this is a limitation, the physical limitation of the rack system. Non-zero default values is interesting because I think we added this after we noticed there was one plate where the model cheated, where it was trying to cram a lot of reagents into a single reaction and it was kind of running out of volume. And so what it did was that it indicated a negative volume around the reagents, and this way it was able to kind of add more things in. And so we made it stop doing that by kind of adding this extra pydantic check. So we also had to do some kind of ranking on the designs that we're sending to Ginkgo. And this is because not all plates that the model design pass a pydantic validation. So we were generating kind of 2x the number of target plates. So if we're targeting 128, we would generate 256 plates. And then this will give us kind of a surplus of plates, and we had to rank them somehow instead of randomly sending them to Ginkgo. So we tried the different heuristics for ranking. I don't think any of them was really super correlated to the outcome, but I'll just kind of talk about what we did anyway. The first one we did was kind of like this vibe grading regime where we had GPT-5 then act as a PI, and we told it, here's an experimental design from a postdoc in your lab. The experimental design was just the play object and the lab notebook. And you can kind of grade it on how good it is, kind of going from a scale of blind unguided search to a very good, sound scientifically rigorous experiment. So that was one way of ranking. The other one was this idea of kind of checking your work instead of the final result, because we don't know what the final result is. So we kind of looked at all the different actions the model was taking on the computer as it was designing the play. And then we had a separate model come in, look at its work and say, okay, did it do kind of the right things that make sense? So some of these are pretty obvious. Did you at least look at the raw files from the experiment? Did you look at the reagent loss to think about batch effects in the reagents? There's also a time point field in experimental results metadata. Did you look at that field? And this really guards against kind of designs from the model where it's actually just a grid search and it's not thinking really hard. So this kind of filters for the smart designs. And I think the fact that this is not correlated that well with the final titers is just maybe sometimes in science you do all the right things, but you don't get the wild results that you hope for. And then finally, GPT-5 wrote some interesting lab notebook entries. I think these slides will be online, so maybe people can read them a little bit later. But it does consider a lot of the biochemistry of these reactions. I don't understand most of these. They kind of vaguely make sense to me. So it thinks about core ions and buffers, energy modules like ribose, glucose, and MPs, different cofactors that it was adding in. I think we had some interesting high titer results with both spermidine addition and also catalase. So the model had this theory that, okay, the catalase is good for consuming free radicals, but also generating additional oxygen at the liquid-air interface. Unclear if that's a real mechanism, but we did have a very good experimental condition come out of this one. So yeah, overall, what we learned was that I think by using the pydantic model to constrain the action space of GPT-5, it actually made very few mistakes. So from the 36,000 different experiments we did, there were only 2 bad plays. There was a negative volume one that I talked about, and one that was kind of silly was a simple math error where it was kind of converting nanoliters to microliters incorrectly. And so it basically accidentally set all the other reagents to 0. And so we had a play that was just a sugar gradient. And then human oversight was still required for kind of improving the protocols and handling the reagents on the off-the-rack system. So we saw that the system can design and interpret experiments, but we see that lab work still requires kind of these practical details, and we do need experienced operators. And kind of looking to the future, we're really excited to apply AI and more of these lab-in-the-loop optimization workflows. And we really see kind of autonomous labs as being complementary to AI models, where the models can design the experiments, but ultimately biology really still requires testing, iteration, kind of validation in the physical world. So yeah, that is all from me. I guess next up is Todd, or are we taking questions?
Oh, no, that's great. So I think Todd will speak, and then we'll do questions at the end.
Okay.
All right, hello, I'm Todd Edwards. There we are. So I am with EMSL, which is the Environmental Molecular Sciences Laboratory over at the Pacific Northwest National Lab. What I'm going to talk to you about today is what we are and kind of what we're trying to achieve and the steps we're taking along the way to get there. So EMSL is a user facility, which is a type of lab within the Department of Energy Office of Science, where we don't do our own research, but we have scientists and equipment, and we partner with folks around the country to take their samples, do whatever kind of analysis they need, and then they get the data for a year, and then the data becomes public. So we're able to build a large public repository of interesting data that hopefully is all done consistently so that it can contribute to larger projects. You can see we have functional systems biology. This is traditional studying microbes in culture. We have environmental transformation and interactions. And this is looking at the interactions between microbes and soil or atmosphere, plant roots. So looking at it in the context, and that's for building Earth system models. And we have a computing group that helps build models and use models in our research. And so there's a QR code here, so you can check us out, find out how you could collaborate with us. And then Science Central is up at the top. That's where you can get access to the data. Now, because we have been very traditionally working individually with PIs, very small scale, specific experiments, but we have a vision where we want to automate in order to improve the rate at which we can generate data and improve the consistency that we're generating data. But we're not doing production, right? We're not doing the traditional high throughput, doing the same few processes over and over. We have to really tailor every time we work with a new collaborator, tailor the experiments to what they need. But we still want to generate that high quality, consistent data. So we need modularity in our platforms, both on the hardware and the software side. And then, as things have grown, autonomous experimentation has cropped up and proven to be a very efficient way to build out the data that we need to make the models or improve models over time. And again, we have access to LIMS, high performance computing, data storage, so that we can generate the data, analyze it, and make it open. The challenge that we're specifically trying to attack here at EMSL is the notion of the genomes is not enough. Like if you understand the sequence of a microbe, that doesn't really tell you enough to be able to predict how it's going to behave. You need to study the phenotypes, and so you need to study the microbes under different conditions, and in order to build models so that you can predict this microbe, this genetic sequence, under these conditions, how will it behave? And that's relevant for industry. This SYN 3.0, this is an example we like to share. Someone has created a microbe with the minimal amount of genes, but still be viable to grow. And 30% of those genes, they don't know what they do. So it's a very large problem. And in terms of understanding what possible proteins might be expressed and what proteins are actually expressed and how they interact. And the problem's just growing. The disparity between the genomic sequences and the knowledge about the phenome is getting bigger and bigger because more and more microbes are being discovered and more sequences are being produced. So our efforts have been really to try to catch up with the ability to generate genomic data. We want to be able to generate phenomic data. Now, EMSL, like I said, on the right-hand side is our traditional approach, and this is where we work with the individual PI We do a very targeted experiments for them. It's lower throughput, but it's really getting them what they need. But because we're generating it, we're able to pool all the data so it can be used for other folks. What we want to do is do that on a much larger scale with this automation-centric approach, but we don't want to go directly from this traditional approach to the high throughput, forcing folks to do a narrow set of experimental procedures. We want to jump from traditional straight to a large-scale R&D. Let's see. So this is our dream. We want to produce we wanted to commission a device that would be able to do a very wide range of possible experiments at high throughput and with that consistency, and treat it like something like a James Webb telescope, right? We want to make it able to do a campaign of science, study a particular thing, and then once that campaign is over and those folks have their data, shift and point it at something else and study a whole new problem. So when we set about this, we couldn't really plan what throughput we needed or what experiments we needed. We focused more on the range of capabilities that we wanted and kind of a capacity that we wanted to be able to do. And then we went out and took bids and came up with a solution which we are working with Ginkgo to produce. And we call it a capability because M2PC is both the equipment that's going to go in the room, but it's also this whole new lab. So we have a whole new building, new support lab space, and a big automation space, plus this rack system. And you can see it may be hard to see from the back, but we have different pods, and each one is focused on a different type of capability. So there's the ability to produce media, so we can do a wide variety of plates. We have a cultivation section, we have analytical tools, we have mass spec, we have sample prep, and then we have a BSL2 section. And so this is all in the design process right now, still settling on what we're going to actually get. But the idea is that instead of now working with individuals and doing experiments on, say, a single mass spec, we're going to work with a campaign. So we'll get a group of scientists together that are all focused on a similar problem, and we'll plan out a series of experiments and then run it on M2PC. And then while that's running, we're planning the next campaign. So when they get their data, we can move on and start the second campaign, and so on and so on. But this is this is the goal, this is the dream. It's going to take a few years. So how do we get there? So in the meantime, we've been taking steps along the way to build out all the infrastructure, all the adjacent things that we need to accomplish in order to get there. And I'll walk you through that. Our first one is automation. Like I said, we have been a fairly traditional lab, not much automation, and we wanted to kind of dip our toes first. So we did a similar kind of commissioning process to contract a system to do what M2PC is going to do, but at a smaller scale and in an anaerobic environment. And so, and we also worked with Ginkgo on this one. And instead of doing racks, they built what are called tiles, which are very similar to a rack, but they're enclosed in these anaerobic pods. So we can do all of the cultivation, the growth experiments, we can do analysis with HPLC and some other tools, and produce plates and do those kind of studies on anaerobic microbes while keeping them in anaerobic conditions the whole way. So we don't have impacts of working in oxygen or trying to get rid of oxygen. And this is also forcing us to figure out how do we manage this kind of level of throughput in the lab that we're not used to. So there's a lot of consumables that have to go in and reagents, and there's a lot of waste coming out, and we need a kind of a new group of people to support it. And so it's better to do this now because we're going to need that when we get to M2PC, we're going to need even more. We also have a computing analytics and modeling team, CAM, I mentioned them before. They have a section called MIDAS, the Modeling Integration Data Agents for Science. And their goal is to build AI agents that will help all along the way. And this is kind of one typical experiment of how we work. Users might come in and they go to the JGI to get their sequencing. And that sequence data can be used to create models, which are not so useful when they first come out because they're not constrained. So they don't predict very well, but we can do experiments doing the phenomics to constrain those models and improve them so they can do better predictions. The goal eventually being to build a digital phenome of the microbe so that we can predict the outcome, the functional parameters of the microbes. And so we're building tools now to help build those models, improve those models with data. And then the next step is to build build agents to help design the experiments that would be run on the systems so that we can eventually get to that closed loop autonomous experimentation where we are using AI to efficiently plan experiments to sample that whole parameter space. Right? Because the growth conditions we saw in the last talk, there are many different variables that you can adjust. A person trying to do that is not efficient, whereas AI can do that much more efficiently, so we can get a model that works. The next thing we're working on is to standardize our workflows. So like I said, we get all sorts of input. Different people have different workflows needs. But from our standpoint, we need to standardize, right? So we're trying to come up with a way to represent automated biology workflows in a standard way so that we have a definition of them, so that we can compare data that comes out of these workflows apples to apples instead of not being able to compare them. Right? Because if your experiment is basically the same as your experiment, but it's different enough that you can't compare your results unless you know exactly how they are different. And as anyone has tried to work with a biologist who has they ask for a cultivation, and then you have to start probing, well, what are all the details? Eventually you can sort it out, but there's a lot of work. So we are building tools that will help us onboard these workflows, convert them into our standard format, and then you convert that standard format into instructions for our automation. And then the last little bit, this is where I've been more involved, is trying to pull it all together. So we've got the automation, we've got the workflow, we got the tools to design experiments, but we hadn't put it all together. And if you've done interdisciplinary work, you know it's not easy. So we decided to do a minimum viable product to show this loop going all the way around, and then forcing the biologists and the systems biologists and the automation people and the coders, the modelers all to work together and understand really what the other teams need and sort out those interfaces. And we were able to do that. So we have a tool that orchestrates this all, and it takes an input file that defines what the experiment is. We have another tool that designs the plates, and then another tool that turns that plate design into pipetting instructions. We used a TCAN Fluent. And we did a growth experiment where we measured the OD every hour, and then took that raw data, got the growth rates, and then did a check. Have we met our conditions to exit? If we have, it generates a report. If we haven't, it takes that data and it adds it in and plans the next round of experiments. And again, this was only a proof of concept. We call it Aurelia because everyone was using MVP for other things. So that is the autonomously repeating experimentation leveraging integrated AI. But here's kind of the short version. So we're using Aurelia lipolytica, which is an industrial relevant yeast. And it grows on a carbon source as well as a nitrogen source. And the nitrogen can be expensive, so the experiment we're doing here is to try to grow on a cheap nitrogen source, which it doesn't grow very well, but if you have a little bit of the expensive one, you can get it to grow. So we're trying to figure out what's the ratio that gives us the best growth while trying to minimize the amount of the expensive one. Now in A, we have to do some manual work to get the cells in the right spots so that we can do the growth experiment consistently. B, it shows the plate map that was designed. We have repeats. We checked the edge effects. And then it came up with these conditions. And then we built the plates, we grew them. And C shows a few of the different conditions that we tested. And you can see some are more consistent than others. But the tool is doing the statistics under the hood and using that to produce D, which is the map of the two different nitrogen sources, and the darkness of the blue says how fast the maximum growth rate. And you can see it didn't have enough information there to keep going. So the red dots are the ratios that it picked for the second round. And at the end, at the second round, you can see there's kind of a plateau in the lower left. And so because we were just doing proof of concept, we said it's done, it found its confidence well enough to produce that, to produce the exit, and it worked. But you can see this obviously needs a lot of improvement, right? It didn't look at anything in the upper right. We could tighten this up. We can show more about how it decided to maybe ignore that one dark spot up in the upper left. But fundamentally it works, and now we are working to improve this tool and also to extend it. So this was done with a Fluent that had a plate reader and a single incubator shaker. Now that we have AMP2, we have access to a larger and better incubator. We have more analysis tools that we can rope into this. So the team is working to expand it. But overall, we're getting there. And it's a process that we need to accomplish by the time M2PC gets here. So this is our grand vision. On the upper side, we've got the green. This is onboarding workflows, getting them into that standardized format. The blue is the platform, so taking that standard format and generating scripts that can run. And by separating those two, we can build these tools out in a modular way. So folks who are focused on the onboarding tools, they don't have to worry about also making it work with the Fluent and with the AMP2 and with every other system. They can just get it to the standard format. And then we got specialists that work with the automation that can take from the standard format and get it down to the scripting level. We have our Science Central where the data comes out. And then Midas on the orange side doing the tools that interact with the models to take the data, improve the models, use the models to make predictions, to plan experiments, and do that autonomous loop. And the last piece I would say about this is that this model side can be as complex as it needs to be. So it could be a simple design of experiment tool, or maybe we can incorporate OpenAI and do a much more complex design, but that still doesn't have to worry about how do we implement that design on our automation. They can just focus on that tool. And so that's our vision. And I realize now after watching Will, I forgot to put my contact information up here, but you can find me at EMSL, Todd Edwards. Find me on LinkedIn. I'm happy to chat. And then, yeah, definitely if you're a data person, check out Science Central and make use of that data because taxpayer money funded it, so it's free. Thank you.
Let's thank Joy and Todd.
It's Ginkgo.